Nalley, and she will be talking about environmental justice through the lens of coastal health. And want to just thank everyone, including Eileen, for accommodating our schedule change. We originally said last week, but there was a holiday. So thank you everyone for being here. So Eileen Nalley is an ocean and coastal ecosystem health specialist with Sea Grant based at UH Manoa, and she specializes in the environmental and human health impacts of marine pollution and shifting land use in the Pacific Islands, with an emphasis on environmental justice. She uses interdisciplinary approaches to address diverse topics in partnership with community resource stewards and multi-stakeholder groups. She earned her PhD in marine biology in the Tobo lab at UHM, where her dissertation examines anthropogenic impacts on diet specialization in herbivorous reef fishes at a range of spatial scales using a diversity of tools spanning metabar coding to meta-analysis. She also worked with Dr. Megan Donahue at the Hawaii Institute of Marine Biology to develop a series of projects assessing land-based pollutants in commonly consumed reef fishes in Hawaii. As a postdoctoral researcher, she continued this research in the Marshall Islands and worked with NOAA to develop stressor thresholds to inform coral reef management. All right, so I'll let you go ahead and take it away.
Thanks, Carrie, and thank you everybody for being here today, and for, as Carrie said, accommodating our shifting dates. So I work at the Hawaii Sea Grant, and I work on sort of a variety of different projects, but all under the broader umbrella of the health of coastal communities and ecosystems, and sort of at the intersection of a number of different fields. And so I'll be sharing with you today a bit about that work, and diving into a few of the projects that I've been working on. But this is sort of an overview, so I'm happy to discuss anything in detail that anybody would like. You'll have my contact information. So anyway, thanks for being here, and I'll dive right in. So I'm originally from the sort of central trading grounds of the Mississippi Delta, Bulbancha, as it's known, home to the Choctaw, Chitimacha, Biloxi, Coshada, Homa, and other tribes. And many now call this New Orleans. But for the past decade, I've been living and working on Oahu in Hawaii, based out of the University of Hawaii at Manoa. And I want to acknowledge that this is land that has been occupied territory following the illegal overthrow and annexation of the sovereign kingdom of Hawaii by the US military and agricultural businessmen in the late 1800s. And this is a really important piece of the story of the kind of work that I do and of the story of this place. So while Hawaii is undeniably a beautiful place, it's also obviously very heavily impacted with significant development along a very narrow stretch of coastline, and a history as well as ongoing story of racism, oppression, and detachment of Native people from their lands, culture, and stewardship practices, or at least an effort to do so. And this affects many different facets of the health and well-being of ecosystems and communities in this place. So this detachment is an ongoing practice largely accomplished through militarization, which has since spread throughout the Pacific Islands, so it's not unique to Hawaii, unfortunately, as well as settler colonialism and economic displacement of people from their place, layered on top of unsustainable rates of tourism, which many of us got to see sort of a different side of the coin during COVID, when we saw bait balls and swarms of sharks coming through Waikiki. Once again, it was sort of a reminder of the impacts of tourism and the density of tourism on people and places here, as well as industrial agriculture, both historically in the form of sugarcane, pineapple, and at present with agrochemical corporations like Monsanto using the land for seed crop experimental studies, so not even growing food, but examining sort of the herbicide resistance capacity with the extended growing season here. So all of these different land use practices have a significant impact on the place and leave a legacy. But this isn't a story that's unique to Hawaii by any means. It's happened around the world. And each one of these activities imprints in a unique way the ecosystems around it, which then result in this transition of coastal ecosystems from looking like vibrant, diverse, healthy, dynamic coral reefs to systems that are far more vulnerable to global stressors like climate change and ocean acidification. They have an increased susceptibility to disease, such as this bleached coral reef. And in addition, they're more vulnerable to local stressors. And this can include unsustainable rates of sedimentation, eutrophication, direct inputs of sewage onto reefs, which is something we see often here in Hawaii, unfortunately, as well as the input of pollutants from carried with the sediment or more directly through water and runoff or groundwater. So these factors on a global and a local scale all contribute to this general habitat degradation and the loss of the capacity of ecosystems to support themselves or to sustain themselves and bounce back from disaster. And this is really important, this loss of resilience, because coral reefs are inherently extremely complex systems. The ecosystems as a whole, but also the corals themselves harbor this community of organisms. Much like us, they have a microbiome of microorganisms, fungi that all contribute to this coral holobiont, which helps sustain this very complex function. And corals themselves then act as ecosystem engineers and provide habitat for thousands of other organisms which grow on and among them. So as these systems degrade, we're not only losing an ecosystem function specifically, but we're also losing the myriad of other organisms that are reliant on this habitat for food and shelter. So it's a cascading series of impacts. And circling back around to directly human issues, this is problematic because reefs and other coastal systems support a diverse array of fishes and other seafood sources that in turn sustain communities. And in the context of environmental justice, subsistence fishing and even recreational fishing communities are some of the most vulnerable populations in the world to these impacts. Many subsistence fishing communities live sort of at the margin of this traditional Western framework of economies and are instead reliant on sharing economies, whereby everyone has access to shared resources. And so when habitats no longer sustain these shared resources, people are left in a very precarious position, not only financially, but this can also result in a lost capacity to pass on intergenerational knowledge of ecosystem stewardship and sustainable harvesting practices. And importantly, this can have really irreversible and tragic consequences for people and ecosystems if you lose this intergenerational knowledge of how to care for a place. So in addition to this potential lost resource and knowledge base, there's also a loss to diverse, essential, nutritious, and affordable protein sources. And so without access to these resources, populations in many Pacific Islands have undergone a nutritional transition, whereby the reliance shifts to unhealthy imported foods. And in these cases, you often see a proliferation of preventable diseases like heart disease and diabetes because of this loss of sustainable harvesting and access to healthy resources. So by addressing these anthropogenic impacts in coastal systems, my work aims to not only restore the ecological abundance of these places, but also support the cultural connection of communities to the places that they care for, so that this knowledge of sustainable stewardship is able to be passed on through generations. And one of the primary ways that I do this and that I've been sort of working towards this goal is through examining land-based pollutants in coastal ecosystems. And as Carrie mentioned, I've done this sort of in a variety of different capacities. And so today I'll touch on a few of those, but I'm happy to discuss them more broadly with anyone who's interested later. So the first step, so to speak, in this series of projects that have been examining land-based contaminants in coastal ecosystems was sort of to establish baseline concentrations, because this work was largely, this was a largely understudied topic in terms of contaminant concentrations in reef fishes. Typically, when we think about contaminants in fish, we're thinking about high mercury in tuna, things like that. But tuna is not what people are catching. Some cases people are catching it from the shore, but often when people are going out and shoreline fishing, easily accessing resources, it's reef fishes that people are eating. And so we really wanted to establish baseline concentrations of contaminants in reef fishes. And so working with Megan Donahue, Julie Zill, Maddie Schmidbauer, Honor Booth, we set out to look at more than 20 different sites on Oahu and Kauai as a start, and looked at around 200 fish samples. We also collected sediment from each of these locations to look at trophic transfer. And our primary assessment was looking at metals, but a subset of these samples were also examined for pesticides and PCBs. And this first pass was really just casting a wide net. We looked at sites with a variety of different land use types, ranging from landfills, sites adjacent to industrial areas, residential areas, harbors, military sites, so a variety of different locations, just to sort of understand what was happening. And after we did this first broad pass, we were able to then take a deeper dive into a few specific locations, working really closely with community groups, stewardship groups, and state management agencies. And this is really where the environmental justice component of this work comes in, because we were able to, in the process of sort of establishing these initial assessments, people came to us and said, hey, we want to know what's happening in our place. And so having the framework to address those questions helps support this partnership and to really address the concerns of people caring for their places. And so we also worked with Sea Grant to host a multi-sector workshop to bring together stakeholders to identify research that's happening and areas for future work. And that's something we hope to do again in the future. So the first place that I want to tell you a story about today is Pu'uloa on Oahu. And so this work was largely led and done by Honor Booth, who's a chemistry master's student, and she also is a Sea Grant fellow. And we were working closely with Sandy Ward at Malama Pu'uloa, and then in collaboration with Megan Donahue and Kim Polinski at the Nature Conservancy as well. And so Pu'uloa, or Pearl Harbor, is probably familiar to many of you because of its pivotal role in World War II, but it has a history that really far predates that. Pre-colonization, Pu'uloa was the breadbasket of Oahu, and people would come from all over the island to harvest in its more than 20 fish ponds in this one area. And in addition to the coastal abundance, there were sustainable stream diversions that supported kalo agriculture and water flow into areas in ways that supported the wetlands and maintained the diversity of the system, but also provided abundant food resources both on land and in the water. Unfortunately, once the utility of this shallow water port was discovered by the US military, it quickly transitioned in terms of its land use. And that's more aligned with the what you see today on the right. So Pu'uloa now is a highly developed coastline with industrial agriculture, as you can see on the far left, replacing what were once these sustainable stream-fed fields. There's also golf courses, waste disposal sites, and dumps. There's, of course, a significant military complex known as Joint Base Pearl Harbor Hickam. And this area is also designated as a Superfund site in the 1990s and remains one today, which is quite a long time to remain a Superfund site. Because of all of this known impact, there's signs posted throughout the area saying not to fish, But in fact, if you go anywhere along this shoreline, there are people fishing. If you go along Waikele Stream, going through the mangrove estuary areas, there's entire communities of people living along that shoreline in the mangroves, presumably harvesting frequently from the surrounding areas and living off of that. And so this highlights a really important element of this work and sort of the outputs of this kind of work, which is that just because you say a place is polluted and you can't fish, doesn't mean people won't fish. So you need to really consider the socioeconomic reality of places that you're working. A lot of the places, the communities along the shoreline are ranked in the high socioeconomic vulnerability index by the CDC, is something I'll talk about a bit more later, but it's a really complicated area. And while we typically, often the spotlight is on the military impacts, which are significant in some areas, there's a lot of other stuff going on as well that needs to be considered. So we worked closely with Malama Pu'uloa, which are the stewards of Kapapapuhi Point Park over on the far west side of West Loch, and Pouhala Marsh. At Pouhala Marsh, we worked with the Division of Forestry and Wildlife. These are two large wetland tracts that remain in this area, and few remain still in the state. So they're very important areas for birds and other endangered species. So I'll just do sort of a snapshot overview of what we found to give you an idea of what we're talking about in these places. So at Kapapapuhi Point Park, I want you to first look to the top right of the screen where you'll see several fishing piers. Again, I want to reiterate this area is technically closed for fishing, but people line those piers every day, all day fishing. And while it's technically supposed to be tossed back or don't eat the fish, you can imagine a lot of that fish is in fact consumed. So in this area alone, you have a lot going on. There's a golf course on the far left. There's Honouliuli Stream draining through it. There's Nihololokaia, which is a fish pond undergoing restoration. There's a mangrove area that now much of that has been removed through the hard work of Malama Pu'uloa. And so in these various areas, we collected multi-increment sediment samples as well as biological samples that were donated by fishers in the area. And in this one spot alone, we found in sediment samples, which are those orange dots, in the fish pond and in the stream, we found arsenic, chromium, mercury, and pHs that were above thresholds that could potentially impact aquatic life. And in OEO samples, as well as in some sediment samples from the fish pond and the stream, we found DDD and DDE, which are organic pesticides, historic pesticides that have been banned since the 70s under the Stockholm Convention, but are still present in the environment. So these are problematic, of course, when we're talking about restoring these places to abundance and warrant remediation. So on the other side of West Loch, we also worked in Pouhala Marsh. And here you have a similarly complex area. You have Waikele Stream in blue draining the far left side, as well as Kapapapuhi Stream in the middle. Again, we took sediment in orange samples and biological samples, which included fish and crabs in white. And we have a large mangrove area highlighted in green there again, as well as an ash landfill and a sort of covered landfill area there in the reddish colors. And so again, we tested sediment and biological samples for a variety of different contaminants. And here in sediment samples found arsenic, cadmium, chromium, lead, mercury, pHs, PCBs, and DDT. And again, I want to remind you that DDT is, if you've heard of the book Silent Spring, it's a big concern for things like birds. And this is a kind of sanctuary for endangered birds in the area. So this is a big problem. We also, in fish and crabs, again, these are OEO and Omilu and Samoan crabs, we found PCBs and DDE in some of the samples taken from this area. So definite evidence of trophic transfer of these organic contaminants from sediment into biological biological organisms in these places, which is of concern for both the ecosystem, the endangered birds, the organisms themselves, as well as the communities and individuals trying to restore this place to abundance. And so by identifying some of these known contaminants and their likely sources when we're talking about sites adjacent to a landfill or historic agriculture, we're able to then get a step closer towards successful remediation and restoration of these areas. And it's really important that we do that in these places because there is so much effort and so much community drive to restore these wetlands, not only just to sort of a status quo, but to restore them to their historic abundance. And this is a goal that should be attainable, and it should be something that we all are working towards. And so, for example, here at Pouhala Fish Pond, which is over a little bit to the east in the larger Pu'uloa area, you have community members working very hard to restore this fish pond, working closely with the military to, all towards the common goal of getting this place restored to abundance. And so in communities where there is already so much motivation, and for those of you who listened to Sandy Ward's presentation a few weeks ago on all the work happening at Malama Pu'uloa, there's so much effort and so much determination to get these places back to this edible abundance that they used to have. And so it's important that we are able to say what's going on there and use that information to move forward rather than just sort of hope for the best and not identify contaminants. But in doing this work, as you can see, it's really complicated. There's a lot going on in this area, especially when you're talking about historic contamination. And so in doing this work, there was a clear need for expansion and next steps. So we worked together with partners, including Malama Pu'uloa, to apply to NASA's first environmental justice cohort of projects. And this has been a really neat project or process because it is their first cohort of an environmental justice program. So we were able to offer a lot of feedback throughout the process. And even just this morning, I participated in an interview for NASA's project program for their environmental justice program to offer feedback on improving their funding process to support this type of work and support engagement of partners in a meaningful way. And so through this, we were able to kind of take the project on a step further. And we're now working on this sort of phase 2, understanding contaminant risks through the stories of Aina and Pu'uloa. And this work is being done with Sandy Ward at Malama Pu'uloa, as well as Mia Camaros at the WRRC, Don Viviani at Leeward Community College, which is situated in the Pu'uloa area, as well as Aurora Kagalo Viviani, who's at WRC in geography, and Brendan Dela Cruz, a GES undergraduate student here at UH Manoa. And so for this sort of next phase of work, we are looking way back to start to understand these legacy contaminants, because as you saw, the pollutants that we were measuring, some of them were, potentially from present sources, lead, PAHs. These can come from the industrial activity in the area, but things like DDT, PCBs, these are not from present use sources. And so we're really looking at historic sources of contaminants. And so what we're doing is pulling these historic land use maps. And one of the benefits for Pu'uloa being so coveted by the military, by industrial agriculture, by other sorts of development, is that it has been very well mapped over time. And so we're pulling historic maps that document land use, as you can see on the left here. This is from 1910. And this red area on the bottom, let me see if you can see my mouse here, this red area, this is all sugarcane production in these historic maps. And then if we move over to 1964, quite a bit later, we see this area has diversified a bit. And so there's still agriculture happening, but there are a number of different land uses in this sort of immediate area. So the goal is to map these land use transitions over time, looking at a smaller scale here. My slide to go forward. There we go. If we zoom in just on the Pu'uloa area, here we have maps from 1964 on the left and 2000, this Landsat imagery from 2000 on the right. And you can see that while there was a lot more diversification in 1964 as opposed to 1910, by 2000, we see there's a ton of different things happening in this area. So we're going to translate these historic maps into GIS layers that we can then analyze land use changes over time. And sorry, I got a little lag here on my slides. There we go. Because we know what kinds of contaminants in many cases are associated with historic land use, with certain types of land use, we'll then be able to develop these contaminant risk profiles based on the land use and the time. So for example, for sugarcane production, and particularly in areas where they had pesticide mixing plants and sugar mills, we know that in the 1910s to 1940s, arsenic is going to be a big concern. So if we see that kind of use there at that time, we note potential for arsenic contamination. From the 40s to 60s, this had shifted to pentachlorophenol and dioxins. By the 1970s onward, they were using more atrazine and amitrin, and each of these different types of contaminants have different associated impacts. And so this is just an example of sort of, if you were to take this one particular type of land use, track it through these time transitions, we can start to develop these contaminant risk profiles, which can help us zoom in on what might be happening in an area instead of continuing to do this sort of broad sweep approach of testing for all kinds of different things in all kinds of places to understand what's going on. We can have a more targeted and focused approach for future work. What's interesting as well is that once we develop these contaminant risk layers, we can combine them things like the social vulnerability maps developed by the Office of Climate Change, Sustainability and Resilience here on Oahu to say, okay, we know these certain communities have a very high risk of being exposed to certain types of contaminants. And they also happen to have very high social vulnerability or socioeconomic vulnerability indices for the CDC versions. So that amplifies the risk because we know that's associated with certain types of health conditions or access to health care and things like that. And so we can start to develop more comprehensive risk assessments for the communities in this area based on this historic land use. And what's exciting and sort of the end goal of this work is to develop tools that can be used by communities for environmental justice work in the future. And so the EPA has an existing environmental justice screen tool that includes things like the proximity to hazardous waste. And this is the Pu'uloa area that I have sort of zoomed in here, as well as particulate matter, ozone, Superfund proximity, but they don't address legacy contamination. And in many areas in Hawaii and elsewhere, this is a big issue with known risks. And so we're hoping to develop risk layers for legacy contaminants that can be incorporated into these existing tools in the GIS framework and allow people to sort of independently assess their own risk and use it to empower efforts for restoration and remediation so that we can continue to work towards these abundant and productive food areas, food security and food resilience throughout the Pu'uloa area. But this issue isn't limited to Pu'uloa. As you remember, I alluded to earlier, we've done this for more than 20 sites around Oahu and Kauai alone. And each place has its own unique history, its own unique suite of issues. And so as we continue to lay this foundation, we'll be able to do more targeted, focused work in a broader suite of locations that can really be led by communities and driven by the desire to know what's happening in a specific place based on this other sort of contextual data that we can continue to build up. And unfortunately, these issues are also present throughout the Pacific Islands. And so while we've done a lot of sort of baseline contaminant work here in Hawaii, we've also had the opportunity to do similar work in the Marshall Islands, working with the Marshall Islands Marine Resources Authority, the EPA, and the Ministry of Health and Human Services to look at water quality in Majuro Lagoon, dissolved contaminants and microplastics in fish, as well as these more comprehensive fish consumption and human health risk assessments. And so this work was done with Megan Donahue, Catherine Perkle in public health, Rachel Dax, John Whitney, Max Signofsky, Claire Lewis, Micah Thompson, Madeline Schmidbauer, and Lauren Arnold. And microplastics work was done by the Center for Marine Debris Research at the Hawaii Pacific University, as well as with our partner organizations in the Marshall Islands. And so similar to Hawaii, the Marshall Islands have a really complicated military history that involves not only US nuclear testing, sunken warships, bombing, but also Japanese occupation and similar activities. This has resulted in a legacy of contamination as extreme as Rinit Dome, which houses massive amounts of nuclear waste and is leaking into the nearby lagoons, as well as heavily populated places like Ibai, which is adjacent to Kwajalein, the military base, US military base at present. And these places, in addition to experiencing impacts from human activities through militarization, they're also experiencing sort of the forefront of climate change impacts and sea level rise. And so as a result of a combination of factors, including not great access to waste disposal processes, coupled with rising seas, in many places you have sort of any kind of debris and rubble used to shore up the seawalls, as you see in this picture on the right. And so this can pose a risk for a number of different reasons. So we worked with the Marshall Islands Marine Resources Authority to identify a number of different taxa of fishes that were of interest for human consumers. Because of their frequency of consumption, and based on our past work on trophic transfer, understanding the sort of range of pollutants that might be present in these taxa. And so we got a suite of fish that represent a variety of different trophic levels that are commonly consumed that could be collected from a number of different places to compare between locations. So in all, 136 fish samples were analyzed for a variety of metals, organochlorine pesticides, PCBs and congeners, as well as PAHs. Fishes were also examined for microplastic ingestion. And the Marshall Islands Marine Resources Authority identified 6 different atolls where they wanted this work done, including Majuro, which has the major population center, Kwajalein, which houses a current US military base, Jaluit, which has military operations and a power plant, Wotje, which has military bases, Utrecht, which has been exposed to nuclear testing, and Rongelap, which was bombed in World War II and exposed to nuclear tests. And so while the focus of this work was not specifically on military impacts in the Marshall Islands, it's impossible to ignore that influence in this place. And in doing this work, several interesting things came out of it, of course, but I'm going to highlight just a few sort of interesting twists. And one was arsenic, which we've studied in Hawaii as well, and we see in pretty high concentrations in certain areas. It was used as a pesticide, as I mentioned, for sugarcane. But in the Marshall Islands, we saw high concentrations of arsenic in groupers specifically. And just to orient you with this figure, which you'll see a couple of, we have arsenic concentration on the y-axis, species of fish on the x-axis. The locations are grouped, clustered at the top by atoll and by sampling site, and the trophic level of the fish sampled is colored, and the key there is on the bottom right. And we have threshold levels based on consumption, serving consumptions, and these are based on sort of average estimates of an average sized meal and an average sized person in the Marshall Islands, and with relative thresholds of 2 servings a day, 3 servings a week, or 1 serving a week. And so you can see here that several sites had these high arsenic concentrations in grouper, and this is total arsenic. And so this is what's typically measured, and the levels are potentially of concern to consumers. But we wanted to take a little bit of a deeper dive because total arsenic is not actually what you're concerned about in terms of human toxicity. It's the inorganic form of arsenic that's really problematic. So we were able to take this step of this work a step further to sort of address the concerns of our partners in the Marshall Islands and examine the relative concentration of inorganic arsenic versus total arsenic. And so here in this figure on the y-axis, you have arsenic concentrations. The circles are total arsenic, which you just saw in the last figure, again, colored by trophic level with the species on the x-axis. And the diamonds on the bottom are the concentrations of inorganic arsenic, or the toxic form of arsenic. And so what we found that was really interesting is that we see this inverse trend where total arsenic concentrations were by far the highest in your higher trophic levels, which would indicate bioaccumulation happening in your groupers, your piscivorous fish, or fish that eat other fish. But in fact, the inorganic or toxic form was highest in parrotfishes and herbivores, your lower trophic levels. So we see this inverse relationship where total arsenic decreases with or increases with increasing trophic level, and the opposite happens for inorganic arsenic. And when looking at this toxic form, none of them were actually above this threshold concentration. So that's great news for consumers for this particular contaminant. And it's aligned with what's been seen by some colleagues in American Samoa. And so this really highlights an important area for future work, especially in Hawaii, as it relates to sugarcane and legacy contaminants from that, because understanding the true risk requires knowing this sort of breakdown and speciation of arsenics. This is something we're looking forward to doing more of in the future to better understand the risk in Hawaii. We also found lead in high concentrations in some areas. In certain sites, again, this is the same type of figure where on the y-axis you'll see lead concentration in fish. Each dot represents a fish that's colored by trophic level. The species are on the x-axis and sites are grouped at the top. And you'll see the FAO safe consumption limit, but it should be noted that there's no known safe level of lead to consume. And what's interesting here is that we see the highest concentrations of lead in the lower trophic level fish, the herbivores. And again, this is something that we have seen other places as well. And so it's kind of an interesting question of what's happening exactly here. And again, merits additional work, but was an interesting thing to note that it wasn't necessarily highest in these heavily impacted militarized locations. Another pollutant I wanted to sort of highlight here are PCBs, which we looked at in all of the sites and samples as well. PCBs would be expected to be associated with military impacts, and in fact, we did see the highest concentrations at Kwajalein Atoll, which has a military base, as well as Jaluit Atoll, which has had a historic Japanese military base. And so this was aligned with what we expected, and in several locations, they exceeded these safe consumption US EPA screening values. And so this is another area sort of highlighting the need for targeted remediation in places with known association between contaminants and land use. And so in some places, like for the lead, we're not totally certain what that link might be. In others, for things like PCBs, if we see them in high concentrations in areas with heavy military history, it's likely that that's the source. And so that can sort of aid in the process of remediation and restoration and working towards cleanup efforts. And again, one thing I want to draw your attention to here is that the high concentrations aren't limited to the purple dots of the carnivores, the groupers, or the red dots, the pelagic fish. We see very high concentrations in some lower trophic level herbivorous reef fishes. And this is something that we've seen in Hawaii as well, where PCB concentrations in fish like pole are comparable to that that we see in omilu or barracuda, your higher level piscivorous fish. And so these organic contaminants can build up in fatty tissues over time. So we're not only concerned about biomagnification, but also bioaccumulation in lower trophic levels, which, to circle it back around to environmental justice and impacts on subsistence fishers, is really important because these are fish that people are eating every day, all the time. It's not necessarily only on special occasions. So it's important that we continue to examine contaminants in these lower trophic level reef fishes because they do in many cases accumulate contaminants in a way that's been unexpected. And just as an overview, we were able to sort of assess these contaminant risks at a variety of different sites and locations, share this back with our partners so that they're able to develop consumption risk assessments from that. But one thing we wanted to do was contextualize our results within the broader scope of the Pacific, because when you're working in very place-based locations, you can say, okay, in this one little area, we see these concentrations in these fish, but how does that relate to other places, other types of land use? And so we complemented this work with a meta-analysis of contaminant concentrations reported in reef and pelagic fishes throughout the Pacific and reviewed over 5,000 abstracts to pull a bunch of data that included 226 species, 66 taxonomic families, and hundreds of distinct pollutants to be able to compare the results between places and start to get us a sense of the general trends that we see happening. And so this is an example using lead. On the x-axis, you can see the lead concentration in wet weight of fish. On the y-axis are taxonomic families, and the families are colored by the functional group or trophic level. So in red, you have your piscivorous fish or fish that eat other fish. All the way up to your detritivores and herbivores, parrotfish, and at the top there. And then I've also included the tissue type. So things like liver would have higher concentrations than your whole fish or your muscle tissue, and those are reflected in the shape of the point. And that dashed line refers to sort of the threshold consumption level. So you can see that there's a variety of different reports of lead concentrations throughout the Pacific. And one thing this also served with is a sort of gap analysis, because we were able to say some pollutants like lead in this example have a lot of data. Other pollutants, we have little to no data on baseline concentrations throughout the Pacific. So it's very hard to compare or assess between locations and see sort of how does your site, how does your community, how does your fish pond fall out as compared to other places. But as I said, our goal was to really use this background data to compare to our place-based work in the Marshall Islands and in Hawaii. And so here I've placed in the black squares data from the Marshall Islands, and in the black circles data from Hawaii overlaid on these Pacific-wide reported values that we synthesized from this systematic review. And you can see that in most cases, they fall pretty much in line with what's been reported elsewhere. And this is good. This is to be expected, right? You typically don't expect outliers, except in sort of extreme scenarios or extreme cases. And so this was interesting to see and important that they kind of aligned well. There are a few outliers here, though, that you can see in the Papahanaumokuakea Marine National Monument, actually at French Frigate Shoals. And you might be wondering, why are there very high lead levels in a monument in a protected area? Nobody lives there. What's going on? And this is just a moment to highlight the fact that uninhabited doesn't equal unimpacted. And throughout the Pacific, there are national wildlife refuges that are the sites of historic military operations, weapons disposal, storage, and a variety of other uses. And this isn't limited to the Pacific Islands. It also includes the Farallon Islands, which was a radio station which typically are associated with high PCB levels, as well as a nuclear waste dump site. And so I want to point towards work done by Kyle Kajihiro and others who are far more well-versed in this. If you're interested in looking more into sort of what's the link between this legacy contamination and conservation, this is a really interesting topic that they've done some great work on. And finally, I just want to wrap it up with some more recent work on Ciguatera in the Pacific Islands. And so Ciguatera is a type of seafood poisoning. It's the most common seafood poisoning in the United States. It comes from toxins produced by epiphytic dinoflagellates that grow on algae or sort of rubble. These are then ingested by fish either directly and accumulate through time, much like we discussed organic contaminants doing, as well as biomagnifying in your higher trophic level fish. And when people eat these fish, you can get a variety of health impacts ranging from mild flu-like symptoms all the way to paralysis, neurological failure, even death in some severe cases. And so I've been working on a sort of pilot study here in Hawaii to set the foundation for this, and we'll step through a little bit of that as well as some future work that we're doing. So again, these dinoflagellates grow as epiphytes, and they're tiny little microalgae. So kind of circling back around to what I talked about earlier, these epiphytic dinoflagellates flourish in degraded habitats in many cases because there's high nutrients, there's a lot of substrate for them to grow on. So while Ciguatera may seem unrelated to land use and coastal health impacts, it's not the case. It all comes back and it's all related. And as I said, the ciguatoxins can either bioaccumulate in lower trophic level detritivores like cole, or biomagnify in your higher trophic levels like omilu, jax, things like that. And that's really similar to the behavior of PCBs. These are PCBs in purple in detritivores, mostly cole, and then in red in your piscivores, your jax and omilu. I'm sorry, your barracuda, kaku, and omilu here in Hawaii. And you can see in the overlapping circle zone, these are areas where the PCB concentration and composition was extremely similar in your lowest trophic level and your highest trophic level in impacted states like Sand Island, LOI, Kaneohe near the Marine Corps base. And so this is just to remind you that there are links between everything. These ciguatoxins behave very similarly to our organic contaminants. So what can we learn through the behavior of those in trophic transfer that can inform dealing with the other issues? And this initial study, though, was less focused on the sort of biological aspects of Ciguatera and more focused on the human elements, what happens to communities, to people who are affected by Ciguatera poisoning. And so I conducted surveys and interviews with fishers throughout Hawaii to understand the impacts of Ciguatera, both financially, health-wise, culturally, socially, nutritionally, and found that in this figure, you can see the number of survey respondents on the Y and the island on the X, and it's colored by whether or not the respondents had Ciguatera. So of the 106 people interviewed, 22% of those, or surveyed, 22% had Ciguatera, which is much higher than is reported to the state. Only 25% of people who had Ciguatera reported it to the state. And the effects of it were, in most cases, 67% long-lasting, lasting more than a week. And so one thing that's interesting to note here is that the respondents roughly mirrored sort of the population distribution of the islands. But you can see that on Kauai, there were a lot of Ciguatera cases as compared to the total number of respondents, almost comparable to Oahu. And so what's happening there is in this map, you can see the points, the size of the points are scaled to the number of cases reported in each location. And up there by Anini and Aina, you see a lot of Ciguatera cases, particularly in Anini, and these were almost all from Kole. And so this is sort of a unique situation that's happening there. Almost all Ciguatera from Kole was reported from this area. And despite this high concentration, most previous work on Ciguatera in Hawaii has focused on Roy, and a lot of it has focused on the Kona coast of the island of Hawaii. And so we see this sort of bias in where we're working versus where these cases are occurring and in the taxa that are causing them. So it's definitely merits more attention, is understudied. And so here, just to sort of emphasize that again, we have reported cases of Ciguatera by island, and the fish responsible is colored by trophic level. So on Oahu, you can see most of the cases were from piscivorous fish, or fish that eat other fish. These included largely knife jaw and uku. There were some Roy, but a lot of people know that Ciguatera is, you can get it from Roy. So a lot of people don't eat Roy because of that. So there were a wider breadth of other taxa. And then on Kauai, you see this large proportion from herbivores and detritivores, and these were mostly from Kole. And so this is really highlights differences between locations that we need to investigate further. And these three taxa were responsible for 49% of reported Ciguatera cases. So instead of focusing so much effort on one species that most people know to be ciguatoxic, like Roy, it's important that we branch out and start looking at other species that people are eating and getting sick from frequently. So this work is sort of being expanded upon through the development of the Pacific Ciguatera Network, which is being supported by PAC-IUS and NOAA's National Harmful Algal Bloom Observing Network, where we're trying to partner with collaborators throughout the US-affiliated Pacific Islands to develop this sort of knowledge sharing hub of Ciguatera data and information and research so that we can work together to address this issue moving forward, because it has devastating impacts on fishers and is largely understudied. And so what are the implications of all of this then for environmental justice? Just to wrap us up, well, subsistence fishing communities are often isolated geographically, which makes them very vulnerable to the impacts of militarization, industrialization, and colonization. And so addressing contaminant risks and the loss of resilience in these subsistence and recreational fisheries is really critical to public health and the well-being of the communities and ecosystems. But it's important that we're generating information that is adaptable and can lead to effective guidelines, because the context is really important, and just a broad ban on fishing is often inappropriate. So we have to use a diverse set of technical tools and close collaboration with communities to answer questions that are of importance to them. And that requires these sort of long-term relationships, collaborations, and care and communication to successfully do the work. And then finally, I just wanted to say something I've been trying to do more and has been received pretty well is providing feedback to funders on how to improve the process and make truly equitable partnerships more possible by adjusting their expectations, timelines, etc. And so with that, I'll take any questions if we have time, and you're welcome to get in touch with me if you want to talk about any of this. We're hiring for some of these projects, so if you're interested, please get in touch, and thank you all.
All right, thank you so much for that really excellent talk. A lot of good information in there. So I'd like to open the floor for questions. So you can feel free to enter them in the chat if you want me to read them on your behalf to the speaker, or you can raise your hand and I can enable your mic. I'll also enable video, so if you want to turn on your video, you'll be able to do that now. So it looks like Aurora got us started. So in the chat, Aurora says, what if activities like illegal or undocumented dumping aren't captured by land use? Do you think that they're relatively small signals, or do you trust that the data are sufficient?
Sorry, I couldn't get to my unmute button with my screen shared still. Yeah, that's a really good question, Aurora. I mean, inherently illegal and undocumented dumping probably are not going to be documented in the things that we're looking at or trying to sort of include in these, in the mapping exercise, if that's what you mean specifically. But in terms of capturing land use, there may be ways to sort of translate some of those potential impacts based on other factors, or that may be more sort of reliant on observation data. And so something that I didn't mention for that is a phase of that project is to take the initial mapping efforts and have a community workshop at Leeward Community College and invite people to offer feedback based on lived experience as its own suite of data. And sort of say, do you know of any other potential impacts here? Do you know of any other land use types? And so maybe illegal dump sites would fall into that, as well as like any things in personal memory that might not be captured on maps or in sort of formal documentation. So I think in that element, we would be really reliant on hopefully the feedback that we get from people participating in these workshops. But that's it. Yeah, it's a good point. And hopefully the output will be a living thing that can be updated as new information becomes available.
Okay, we've got two more questions in the chat.
Okay,
one
let me
is
see
from
Nicola's.
Yeah.
Do you want me to read it? What are
Yeah,
some
that'd be good for the
remediation
recording.
methods? Yeah, what are some remediation methods that you think could be effective for restoration of Pu'uloa? Thanks, Nicola. That's a great question. And I'm not an environmental engineer, so I do not have the answer to that, but I know that there are a lot of really exciting technologies and options out there. I think Sandy and others have, explored some of those for the potential future use. And yeah, that's where maybe a whole different sector of partners needs to come in and be creative about environmental engineering and remediation efforts. It's definitely possible. And I like to think of all the magnificent things that we can do as scientists, humans, and engineers. So I refuse to believe that remediation of Pu'uloa is beyond the capacity of our collective intellect. So I don't know, but somebody does, and we just need to find them. Sandy, I saw you had a question. What would you ask of this group to work with the DoD to create improved procedures for unobstructed access to water, sediment, biological sampling in the nearshore waters of Pu'uloa? Yeah, I mean, it's the people's water. So I think, there should be guidelines for how to sample in there appropriately and without jeopardizing any sort of security measures. As I said, there's a lot of activity along the shoreline already. So I think just finding ways to do that in a way that everyone can feel comfortable with. And yeah, but obviously work needs to be done. A lot of work needs to be done in those waters. So it's an important thing that we need to sort of formalize the procedures for moving forward, I think.
I just want to commend you for your perseverance. I know we worked with you to try to create that Hawaii, but I would love to see every researcher not have to go through the same ordeal. So just calling on everyone here to work together on that. So, and just since they unmuted me, I'm going to applaud in person.
Thank you.
Okay, we're technically after the hour, so I think I'm going to close it up for questions unless someone has a really burning one that they want to slip in at the last second. But yeah, thank you so much to Eileen for presenting to us and joining our seminar series.
Thank you. Have a good afternoon.
Have a good weekend, everyone.